Okay, so let's talk about the eye and how it collects visual information. So some of you may have studied eyes in the past, and maybe some of you have even had the pleasure of dissecting an eyeball. Very exciting. So this might be a bit of a review, long story short. But okay, so we've got a candle out in the environment that the eye is looking at and collecting information about. Okay, so that information is first going to pass through the pupil. The pupil is actually a hole in the musculature of your eyeball that allows light to come into your eye. It's probably the one structure that everybody in the world knows the name of in the eyeball, because everybody knows that their pupils dilate when it's dark out and that they contract when it's really bright. Your pupils can dilate and contract like that because of the action of the iris. The iris is the colored part of your eyeball. It's blue or it's brown or it's gray or it's green, or if you're Elizabeth Taylor, it's periwinkle, whatever. Your iris is actually kind of interesting because as depicted in this picture, you'll notice that it's brown. It's not depicting a brown eye, it's depicting a brown iris. The muscles of our iris are brown naturally, and the muscles are what contract to dilate the pupil and then relax to constrict the pupil when the light's really bright. So if the muscle of the iris is brown, then why do a lot of people have non-brown eyes? There are little filaments that are laid down on your iris during fetal development that reflect light in different light waves, so that some of us have eyes that appear blue and some appear green and some appear brown and so on. Even brown-eyed people have brown filaments laid down on top of their brown muscle. Now, in case you want to test my theory on this, or my claims, I should say, because it's not my theory, you can look at people's eyes, and you'll notice that some people have maybe lighter colored eyes, blue or green or something, and you can see a brown spot in their iris. And the brown spot a lot of the times isn't as reflective as the other filaments in their eyes. You're probably seeing their actual eye muscle, their actual iris, which is kind of weird to think about. But those filaments are laid down during fetal development like somebody took a handful of spaghetti and then spun the iris really fast and got all those strands of spaghetti to sort of point outwards. No two irises are the same. Your two irises are not the same. If you have an identical twin, that's the one thing that for sure is not identical between the twins. Sometimes identical twins will have the same fingerprints, but they never have the same iris, because irises are literally made through this bizarre dropping of filaments on the iris and then this centrifugal spinning thing. Okay, well anyway, I just thought you might find that interesting, and it's not in the book. I just thought I'd share some interesting eyeball stuff I found out. Okay, so the iris controls the size of the pupil. The pupil is what determines how much light passes through the lens. So the lens is that next opaque structure, and it is what either bends to become more curved or relaxes to become straighter to make sure that the light is focusing properly on the back of the eye. As we age, the lens gets more rigid and less flexible, and that's why as we age, people tend to get more farsighted. If you're nearsighted, it's not usually due to age, but farsightedness is associated with an aging lens that has become more rigid. Now, the lens needs to focus the light on the back of the eye, which is lined with a bunch of receptor cells that make up the retina. So the retina is one name for the combination of all the different cells that are collecting light on the back of the eyeball. Now this picture is a dissection, front to back of the eyeball, but you got to remember that the retina covers the eyeball's sides. It's like lining the whole inside of this globe, which is your eyeball. So you have receptor cells all over your eyes. That's what allows us to have peripheral vision and things like that. That light can come in from a very sharp angle off to one side or the other, and it'll land on retinal cells that are on the inner lining or the outer lining of your eyeball. So you've got retinal cells all over the place. Now within the retina, if you go straight back from the lens, there's this particular spot called the fovea, which is where whatever you're focusing on at the moment will land. The thing about the fovea is it's got the best, most detailed vision. And so whatever you're specifically looking at, you'll see in really great sharpness and detail and color. Things that are in the more peripheral vision will land in other parts of the retina and not be processed as well. The last structure to highlight in this picture is the blind spot, where you can see that veins and the axons from the cells that make up the retina are going to exit your eyeball. And in that little spot, there are no receptor cells to collect information in. So each of your eyeballs has a blind spot in it where no information can be collected. Okay, so those are the main structures of the eyeball. The information gets collected by the cells in the retina and then sent up to the optic nerve, where then it's going to get processed, and then perception will occur as your brain tries to figure out what you're looking at. Okay, so I wanted to show a little bit more detail about the retina. Sorry, this picture is a little bit more blurry. Trying to figure out why it's so blurry, but anyway, that's not just me being farsighted, right? It's blurry for everybody, right? So we have this different angle on the retina so that you can see the light coming in and then landing on the fovea, and we're going to zoom in on that fovea, on the retina, I mean. Okay, so you can see as you zoom in on the retina, there are actually 3 layers of cells that the light has to pass through before it's actually finally processed by the rods and the cones at the very back of your eyeball. It's, again, one of those examples. We started talking about this back in Chapter 2 when we talked about how information crisscrosses the corpus callosum and goes as far away from the collection site as possible. It's true even at the level of the eyeball, because information comes in through your pupil. It has to pass through the body of the eyeball, which if you look at this eyeball, you'll notice between the lens and the retina, it's full of gelatin-like substance called the vitreous humor, that it is just one more thing that this, the information has to get through to get to the back of your eye. It's like we're designed to have as much difficulty as we possibly can to process information. So, and then even once it lands at the back of the eye, after having successfully passed through the pupil, the lens, and the vitreous humor, then it still has to pass through the layer of ganglion cells, bipolar cells, and then finally get processed by the rods and the cones. Now these rods and cones are so obvious in this picture, right? The rods are the green ones, the cones are the red ones. The rods are basically drawn very rod-like. They look very straight, and the cones look more cone-like. In the real eye, it's kind of hard to tell the difference between a rod and a cone. They are not color-coded, and some rods look kind of blobby. Even in this drawing, you'll notice some of them look kind of wide, and some of the cones look kind of narrow. So for a long time, researchers who were examining the eyes of different species thought that, for example, dogs only had rods. They didn't recognize that a few of the light collecting cells in the back of a dog's eyeball are actually cones. It's really, so long story short, if you ever dissected an eyeball, you know that it's really difficult to tell the different parts and structures. Our eyeballs, our retinas, contain rods which process black and white information, light and shadow, brightness and darkness. And then we also have cones, which process color and fine details. Here's a nice zoom in of an actual group of rods and cones, and the rods in this picture are the greenish colored things, and the cones are the bluish colored things, which again were stained that color. This is not how they exist in your actual eyeball. And then, ooh, look at that fluffy stuff underneath them. It looks pretty gross, doesn't it? It kind of looks like something going on a seabed or something, doesn't it? The rods are the greenish ones, and the cones are the bluish ones. So see, I color-coded my labels for you. Rods are going to handle black and white, like I mentioned. They're also responsible for detecting movement. One of the things that's really important for our survival is our ability to recognize if something's moving, particularly in our peripheral vision, something that we weren't really paying attention to. That's something that will attract our attention, is if we detect movement, particularly in our peripheral vision. We have a lot of rods devoted to processing peripheral vision. One of the nice things about rods is that they work really well even in dim light. So generally, animals that hunt at night have a lot of rods. That's another reason why they thought dogs didn't have very many cones or any cones, was they assumed that they were nocturnal hunters. Turns out dogs aren't actually nocturnal hunters. They're more dawn and dusk hunters. But anyway, cones, on the other hand, are in charge of processing color, sharp detail, and they require bright light to work effectively. All you have to do is reflect on experiences where you've been out in a dusky situation or after dark, and how you might have mistaken one color for another, to really appreciate that your cones don't work very well after dark or in dark-ish situations. If you've ever gone out stargazing and you've been looking for, one of my favorite things to do in the summertime is to look for satellites as they pass overhead. And the best way to see a satellite is to sort of just point your face upwards and then wait for a satellite to move in your peripheral vision. Your peripheral vision will pick up the satellite much more quickly than if you're actually staring purposefully in the area of the sky where you expect a satellite to appear. What's funny is if you detect it with your peripheral vision and then turn your eyes to look at it and try and use your fovea to look at the satellite, sometimes it'll briefly disappear. And that's because your fovea is really densely packed with cones. Your point of fixation has lots of cones for processing the color of items as well as the sharp detail. So when you first look over at the moving satellite and it lands on your fovea, where there aren't very many rods for processing dim light kinds of things, it may literally feel like it disappeared for a second. You can't process the information. And then as it moves some more, it'll land back into your rods, and you'll be like, oh, there it is. Move your eyes again. Wait, where'd it go? That's one of the things about our cones. They're really great for giving us really great detail, but they don't work very well in dim light. Now that blind spot that I pointed out earlier is made up of the spot where your blood vessels and then all the axons coming out of your ganglion cells and then going off to the brain. So you actually don't collect information. So if you close your left eye and fixate on the dot, the car will entirely or partially at least disappear for you, depending on the size of your screen, your distance from it, and so on. You have to, now you'll notice it's not complete, for me the car's not, I'm playing along too, I've got one eye closed. The car's not completely disappearing. I can still, I'm aware of parts of it still being there, and I'm not sure if that's actually my eyes sensing the details of the car or my brain's perception. Like I'm imposing meaning that the car is still there. I'm not exactly sure whether I'm actually seeing it or I'm inferring it. But here's the thing. I am collecting some information about that car, I think. If I open my left eye, of course, I can see the car even if I'm fixating on the dot. With both eyes open, I definitely can see that there's something. I'm not sure, if I didn't know that it was a car, I'm not sure that I'd know that it was a car. Now that it's hard to say, there's definitely something over there. Our blind spot on our left eye is in a different position, is on the opposite side of our eyeball from our blind spot on our right eye. So usually one eye compensates for what's missing from the other eye. So for most of the time we don't notice that there's a blind spot, because when it's landing in the blind spot of our right eye, like it is when you close your left eye and stare at the dot, the car falls into the blind spot of the right eye. With both eyes open, that car just goes ahead and falls in the normal part of your eyeball on the left side. So usually one of your eyes is picking up the information. Also, usually we're moving our eyes around on a scene, and so we don't notice that part of it, had been absent before and we hadn't been noticing it. And then also, if that was, let's say in real life, a car, and let's say in real life we're blind in our left eye, and let's say that in real life we're walking down we're about to cross the street, let's say, and we're looking straight ahead of us at that dot, and there's a car coming at us from the right. Let's pretend like this is a real car that's moving. Movement would attract our attention with our right eye, so that some of our rods would activate us to turn our heads. Hopefully. I mean, obviously, if you're not paying attention and things like that, people get hit by cars all the time. But usually our visual system is designed to attract our attention to movement and things that surprise us and stuff like that. So generally you don't notice the blind spot, and actually it can lull us into a certain sense of security, which is probably why sometimes we step off the curb when we shouldn't. Oh, I did a little depiction. Oh, that was cute of me. Look at my little animation. That's so cute. Okay. Now the last thing I wanted to mention about how the information leaves our eyeballs is the optic chiasm. So as your optic nerve leaves each eyeball and enters your brain, it first crisscrosses at the optic chiasm before being sent by the thalamus to the proper part of the visual system. So there's that X in the middle of your brain where the nerves crisscross. For a really long time, ancient researchers 150 years ago or so, when they were dissecting brains and eyeballs for the first time, they tried to figure out if those nerves are crisscrossing, why is it that if you lose an eye, you can still see it in both visual fields? And why is it that if you have a stroke in your left hemisphere that damages your left visual cortex, you can still see with both of your eyeballs? You know, like, why? They couldn't understand what was going on. It wasn't until they got electron microscopes that they realized that at that optic chiasm there isn't complete crisscrossing of the optic nerves. Half of the nerves from the left eye and half the nerves from the right eye end up in the left hemisphere, and half the nerves from the left eye and half the nerves from the right eye end up in the right visual hemisphere. So you don't have complete crossing, you have half of each eyeball. So what we end up is what I talked about back in chapter 2, which is the left visual field lands on the right side of each eyeball and therefore lands in the right visual field. Visual cortex. And the right visual field lands on the left side of each eyeball and ends up in the left visual cortex. So that's how the information passes through, and that's how you end up with still being able to see with both eyeballs even if you have damage to one hemisphere or something like that. There's partial crisscrossing. Alright, so we already used the apple and the pencil in chapter 2, so that should look familiar to you, that left visual field goes to right hemisphere and right visual field goes to left hemisphere. Okay, let's go ahead and take a break here and we'll come back and pick up with color vision.